Okay. So looks like we have a great crowd today. I'm sure they are eager to hear what you have to say. And we will start the lecture. Welcome to our international lecture series on bioprocess engineering. Today, we will enter the sixth session of this lecture series with a very interesting and relevant theme in photobioreactor and application. And this session will discuss various aspects related to photobioreactor. Okay. In the photobioreactor, and then starting from introduction to photobioreactor, basic photosynthesis and algal growth, types of photobioreactor, component of a photobioreactor, key design consideration, operation modes of photobioreactor, application and case studies, and advantage and challenges, recent innovation and trends, and the conclusion and future direction. So we are fortunate to be able to present Mrs. Angana Chaudhuri and Magister Science, and she is a PhD candidate from Washington State University, and we will share the resume. So this is the resume from Angana Chaudhuri, and she is a PhD student in biosystem engineering with focus on bioenergy, bioproduct, and bioprocess engineering. And then education in Washington State University. She motivate and accomplish research with a robust academic foundation and extensive experience in biosystem, chemical, and energy engineering. And currently pursuing a PhD biological system engineering at Washington State University and specializing in bioenergy and bioprocess optimization, proven expertise in laboratory tech skill and software and language, and then have a lot research experience, and then the publication and adept in driving innovative solution in bioenergy, waste management, and sustainable bioproduct, receive a prestigious academic and research award, and demonstrating excellencies in leadership and technical proficiency. And with her academic background and the experience, she will share in depth insight regarding innovation, challenges, and the latest trend in the field of photobioreactor. And we hope that throughout this lecture, we can all burn on our horizon and discuss and then gain inspiration to develop bioprocess science, especially in the application of photobioreactor technology in the future. And then thank you for your presence and the participation. Let's listen together to the presentation from our research person today, the speaker. And then it's your turn, Mrs. Angana, so you can share your presentation.
Yeah, thank you for the introduction. I'll share my screen. Can you see my screen now?
Yes, we can see.
Yes, we can see it.
Okay, so good morning, everyone who are present in Indonesia and to everyone else who have joined from different places. So today I'll be talking about a short introduction about the photobioreactors. I'm Angana Chaudhuri. As she has already introduced, currently a PhD candidate at Washington State University in the
Sure.
Department of Biosystems Engineering. And I have done my bachelor's in biotechnology, and I have done one of my master's in energy science and engineering and another in chemical engineering, which motivated me to start my PhD in biosystems engineering under Dr. Ehring Brigetta. So let's start with what is a photobioreactor. The term photobioreactor can be said divided into two parts, photo and bioreactor. Now, from the word photo, we can understand that it has something to do with light, and bioreactor is something which we can consider a reactor, which can biologically produce something in a form of biomass or a bioproduct. Now, this photobioreactor together means that a reactor where we are growing some microorganisms which can utilize the light and help in production of some biomass and bioproducts for the purpose of the industry and for the society. So we can say that the photobioreactor is defined as an enclosed and illuminated culture vessel which can be designed to control the biomass production by adjusting the operating parameters. Now, this photobioreactor has a huge area of application. Nowadays, it's being used for sustainable fuel production, wastewater remediation, carbon dioxide fixation, and finally, the valuable products. Among valuable products, you can get like you can get different pigments like the chlorophylls or the carotenoids, and even some of the omega-3 acids and other fatty acids as well, which can be used in the pharmaceutical and in the cosmetic industries as well. Now, we can ask oneself that what kind of microorganisms can grow here. And particularly here, we can see that it is either a microalgae or a cyanobacteria, or in other words, something which some of the organisms which has chlorophyll in their body or in their cells, and it can utilize the light. So what happens is we can see in this cycle that the when in the presence of light and carbon dioxide, the microalgal cells undergo photosynthesis where it enters the Calvin cycle. Here in the Calvin cycle, they take up the NADH and the ATP and at the same time produce hydrogen gas. From this Calvin cycle, this is inside the site happening inside the cytosol, it produces a glycerate 3 phosphate. Now, this component is very important as it can transfer into starch by the process of gluconeogenesis, and from the starch it can produce glucose, maltose, and other alcohols and organic acid. In another pathway, it can produce the acetyl CoA, which can again go into fatty acid synthesis to produce alkanes, TAGs. TAGs are triacyl glycerols or lipid bodies and alcohols. Otherwise, the acetyl CoA can also go into a TCA cycle where it can undergo amino acid synthesis for the production of the protein. Now, whichever purpose we want to engineer our microalgae, for example, the biodiesel or for wastewater remediation or any other process, we will tune them by providing the nutrients which can allow them to follow that one particular pathway. For example, if you want biodiesel from it, it means that we need to produce it in a way where we get more lipid bodies. In those cases, we give a source where there is a large number of fatty acid molecules, and it can undergo the fatty acid synthesis and produce the lipid bodies. Now, these are some of the images where if we can see that the microalgae, microscopic structure of the microalgae. This is a cyanobacteria where we can see the strings of the cyanobacteria, and these are also green in structure. And this is the microalgae which we get in our environment. Now, the most interesting thing about the algae is that it can grow in any kind of environment. It does not exactly need only one kind of media which we can use it inside the lab or in particular conditions. It can even grow in the wastewater given it has the enough amount of nutrients and carbon dioxide for them to utilize. They are kind of like plants where they can taking the carbon dioxide and release the oxygen at the same time. Now, the growth of the algae is also very much like the bacteria, if we can say. So it begins with the exponential phase, it goes to the linear phase, and finally to the stationary phase. So the microalgae, when you're isolating the microalgae from one of the wastewater, you you culture it a multiple number of times unless you get a stable amount of the microalgae, and then you can see that in your growth curve you'll find the exponential phase, the linear, the or the logarithmic phase, and finally the stationary phase. Now what happens is they take up the carbon dioxide, and it undergoes all the cycles that I have told in my previous slides, and finally they release the oxygen. Now, the algae can be grown in different ways, either in an open system or in a closed system. Now, in the open system, it can either be an open pond or a circulating lagoon system, or a slanting photobioreactor, or in a closed system where they're inside a reactor, which can be a stirred tank reactor or even algal biofilm reactor. Now, first we can discuss about the open photobioreactors. Now, this diagram on my left, it shows how they are being grown in an open system. They can either be in a pond system like this, or in an inclined system, or even in a stepwise system like this. And all there are other ways where they are forming like a circular lagoon and triangular ones as well, where they can operate under the open environment. So anything which is not covered is called the open photobioreactor over here. But this also has some challenges like contamination and carbon dioxide utilization efficiency, because in these systems there is not enough of the mixing or the gas transfer involved. It is happening very like naturally. Now, for the contamination, the solution is that we can select a particular environment, use chemical agents. Culture condition can be separate, and restraining devices and multi-stage processes. And for the carbon dioxide utilization, we can vary the temperature, we can manually turbulate it, or even have a high pH for the system. But this system has an advantage which is better than the closed reactors. That is, it has low construction cost as it is not under any particular reactor or anything of that sort. The capex and the opex is very much low, and it is also easy to operate. At the same time, it can utilize the natural sunlight, and we do not have to provide it with something separate like a continuous monitoring or those sort. And it also can move up to large scale productions as well. Now, when we move to the closed bioreactor, here are some of the images where this is like a rotating, like a tubular bioreactor, and this is like a rotating algal biofilm reactor. Now, there are the closed bioreactors are ones where we are using it, producing it inside the bioreactor, where every parameter like the temperature, gas, pH, everything is controlled manually by us. And thus, it has some advantages, which is like the controlled environment and can reduce the fluctuations present. It can allow us to have higher biomass productivity and also help us in utilizing more of the carbon dioxide. It also helps us in utilizing a wide range of microorganisms. Like in some of the recent studies, people have been doing algae bacteria consortium, where the bacteria along with the algae helps in increasing the biomass productivity. And then finally, it can operate year round and in morning and night, as it is utilizing artificial sunlight. Now, the disadvantages of this is that it has high capital cost with operational complexity. Secondly, fouling and maintenance issue. Thirdly, the energy intensive and limitations for scalability. It also has overheating issues, and the gas exchange is quite limited. But the gas exchange is always like one of the challenges that we need to solve in all of the bioreactors. So there are innovations every day and new trends coming up to solve these kind of problems, where we can and have a better way to mix the gases or have a better nutrient supply. Now, in a photobioreactor, what are the components and the key design? So, most importantly, we should have a light source, gas exchange system, temperature control, and finally a mixing system. Here, this diagram shows a photobioreactor which has some of its parts, like it is always under a fluorescent tube so that the light can penetrate inside it. And then you have a carbon dioxide and air flow, and a meter to always measure how much of the carbon dioxide is entering into the bioreactor. And at the same time, you have a collection valve where you can collect the samples from here, and also an agitator which helps in mixing your entire nutrient and supplements for the algal cell. Now, in the photobioreactor, what's most important is that the algal cells get a good amount of light and carbon dioxide. So it is important that it has gas transfer. So the carbon dioxide, it enters from outside in a bubble, which is the gas phase, and then this goes into the microalgal cells, which is the solid phase. And the more amount of carbon dioxide available to the microalgae, it can utilize them to produce more of the biomass. Now along and so this carbon dioxide is one of their like electron source, but for them to grow and multiply they need other nutrients like the nitrogen and phosphate, which they get from the media. And that is also why an algae can grow in wastewater as well, because in wastewater we know for coming out from the industries it has high amount of nitrogen or phosphate. And this so the microalgal cells can get the nutrients uh, from both the gas phase and also the media phase. So, in the key design, we have to keep in mind that the mixing and fluid dynamics is uniform for them, and the material of construction has to be transparent and durable so that it can it can like help in getting enough the light as much as possible with high efficiency. Now with these, the closed and the open bioreactors, they have some recent innovations as well, which are being used. This pie chart shows how they are using horizontal tubular bioreactor, helical tubular airlift, bubble column, and cone shaped and membrane, plastic bags, flat panel and plastic bags. Now, among these, we can see that the horizontal tubular is one of the is getting used recently a lot. But at the same time, a biofilm bioreactor is also something which is getting a recent like attention. And all of these can be sorted according to their capex and opex and advantages. So as we are using an algae-like organism, and in a world where we are trying to get more and green energy, it is important for us to understand the techno-economical aspects, and that's why it's important to know which of these recent innovations and trends can help us to get lower capex and opex as compared to the conventional ones which we already have. For example, a biofilm rotating bioreactor, the advantages would be that it has an higher efficient density cultures, reduced contamination, and ease of biomass harvesting. And the capex is quite low to moderate, and opex will be moderate to high. And the membrane is high. It produces very high efficiency and can have due to I can have higher retention time as well. However, the capex of this is comparatively high, and even the operational cost. And you can understand it why it would be, because it works on membranes, and we are still in a world where the membrane cost is much higher as compared to the other ways where we use chemical ways we use for separating stuff. Now, another way is a flat panel. It has very simple maintenance, but it has some moderate to high capex. The stirred tank reactors has excellent mixing, and but however, it has relatively high capex and opex. The helicular tubular one is also the same. So among them, we can say that the biofilm bioreactor is one of those innovation trends and supplies which is getting more attention because the cost is quite low as compared to the other ones. Now here are some structures we can see. This is the stirred tongue bioreactor. This is a triangular pyramid shaped, and these are the triangular ones. And this is an algal biofilm reactor. This can be a plastic bag. The plastic bag is a very a cheap one. However, they are not good for or not good for aeration and agitation. Now, when we come to so these recent trends are focusing on some things important, like the modern PBRs focus on the improving the light distribution and using optical fibers, LED panels, or reflective surfaces. Secondly, the enhanced designs ensure maximum photosynthetic efficiency in dense cultures, overcoming shading effects. Now, they also have a new process which is called the electroporation, which are enhancing the lipid extraction in the microalgae for the biofuel production. Finally, these PBRs are being optimized so that they can capture the carbon dioxide as much as possible from the industrial emissions. The microalgae grown in the PBRs absorb the carbon dioxide, and hence it is a part in which we can do have a carbon capture and reduction of the greenhouse gases as well, while producing sustainable, valuable biomass for us. Now, these PBRs are now integrated into these wastewater systems as well, utilizing nutrients from effluent streams for algae growth. This dual application cleans wastewater and produces biomass for biofuels and fertilizers. Now, depending on these recent trends, we'll see some case studies which have proved some really good results. For example, in one of the stirred tank reactors where an algae has been used, here along with the carbon dioxide and air in a closed photobioreactor, there has been an increase of 40% increase in the lipid production when compared to the conventional method. So we can say that the stirred tank reactor is quite efficient for the lipid production. Now, this lipid that you're getting, it can be converted to a biodiesel. This is a tubular rotating biofilm reactor where there is a 90% efficiency and reducing the cost for commercial scale food additive production. So this bioreactor has been used for production of the nutraceuticals. We already know that algae can produce some of the like food for which can be used for the human consumption and even for the animals. So for that as well, they are using these bioreactors, and the more amount of light efficiency for these kind of reactors, the higher is that they can produce the biomass that can produce the food additives over here. Then finally, these bioreactors can, since this algae can grow in any kind of media, a wastewater, when it grows in the wastewater, it takes up the nitrogen and phosphate from the wastewater. So when they're taking up those, it reduces the COD, BOD, phosphate, and the nitrogen component. What is the COD and BOD? It's basically the chemical oxygen demand and the biological oxygen demand. When there is effluence from the industries, they have high amount of nitrogen, phosphate, and other components. The oxygen amount in the water itself decreases a lot. So what this algae does is they take up the carbon dioxide along with the nutrients from these places, and it rejuvenates to produce more of the oxygen, which is again get dissolves into this water. And I myself have done one of the projects where we have used algae and bacteria consortium, where we have seen more than 90% of the decrease. Hello,
Yes.
90%. Oh, I thought somebody was speaking something. Okay. Um, and that causes like a 90% of nitrogen or phosphorus reduction as well, which helps in the nutrient recovery. And when these gets recovered, they can be used as the biofertilizers as well. So you don't need a lot of manure or other artificial chemical substances, which might be, like hazardous for the plants as well. So finally, coming to the conclusions and the future directions, we can say that these can be used for the wastewater treatments and the generation of the high value products, and their design and operation enables precise control of the growth conditions and for photosynthetic organisms like algae and cyanobacteria. And they also help in optimizing the productivity and minimizing the contamination risk. While challenges such as high cost, light distribution efficiency, and contamination risks remain, the recent advances in the reactor design and automation helps in providing a pathway for the large scale applications. Nowadays, since with the CRISPR-Cas and other genetic engineering pathways, you can design an algae which can suffice to your needs as well and produce more of the lipid oils or even better for the wastewater treatment. Now, looking ahead, the photobioreactors have the potential to revolutionize the industries for producing renewable energy as well as reducing the greenhouse gases. With this ongoing innovations, the photobioreactors can contribute significantly to the environment, sustainability, and also hence production of the bioproducts. Thank you. So if anyone has any questions.
Yes, we go to the Q&A segment, and then we have another participant here raising hand, like Maura Azra.
Okay. Good morning, Mrs. Angana. First of all, let me introduce myself. My name is Maura Azra Sabrina. I'm from the Agro-Industrial Technology major from Pajajaran University. And I have a question to ask. Is there any disadvantage or challenges of using photobioreactors for cultivating microalgae in the agro-industrial sector, particularly for sustainable biofuel production or nutrient recycling? Thank you, Miss.
Yeah, so these photobioreactors are like still controlled and they are still under the it is getting optimized condition. So it is not like we have started using it in a very industrial scale for biofuels, particularly because of the high capital and operational costs. So like your temperature control, aeration, and everything, and also the materials that you need can cause a high capital and operational cost. But there is one reactor, which is the rotating biofilm reactor, which is already getting established here in US already, which is being used for the wastewater treatment. So they have used an algal biofilm technology to reduce the cost, but they are targeting mostly for the wastewater remediation. So for biofuel production, there is still a disadvantage of the high capital and operational costs.
Okay, I understand the answer. Thank you so much for answering my question, Miss. Thank
Yeah.
you.
Okay, thank you, Maura. And then next, Ikhlas Ragayandra.
Hello, Miss. My name is Andra from Pajajaran University. First of all, my question is actually kind of already answered because I was actually going to ask about what is causing these uh, high cost of creating and running these photobioreactors. But um, since that's already kind of answered earlier, I guess I kind of want to ask instead, is it possible to kind of like produce biofuel using photoreactors in a, like, let's say in an industrial scale where it is possible to compete with traditional or fossil biofuel?
I mean, there is a chance that is because for algae, the substrate which is being used is completely low cost. Like the microalgae does not need a lot of high maintenance and everything. And at the same time, the substrate it is growing in. I mean, if you just replace the media with wastewater, then I think the cost might decrease in the initial stages, so that we can like upgrade it to an industrial scale later on, so that we can get biofuels from that. But at the same time, we have to there is another challenge, which is like the uh, production of the lipids from the biomass. The efficiency or the yield is quite less as compared to the other processes. So if we get a technology which can increase our yield, then we can surely get in we can upgrade it in an industrial scale.
So ideally, if a photobioreactor biofuel is willing, has a chance to compete with fossil fuel, what needs to be changed? Or is there a certain technology that needs to be invented in order for it to compete?
I think the biomass productivity, the lipid yield has to increase to reach up to that level.
Oh, okay, thank you.
Okay, thank you, Ikhlas. And then this from Kanza Putri. Go ahead for your question.
Okay, good morning, Miss Angana and Miss Aisyah. First of all, I apologize for not being able to turn on my camera. My name is Kanza Putri Kairunisa. I'm from the Agricultural Industrial Technology at Pajajaran University. As you have explained about the photobioreactor, I would like to ask what are the latest innovation in photobioreactor designed to enhance energy efficiency and productivity, and how can photobioreactor technology support climate change mitigation efforts such as carbon dioxide capture? Thank you, Mrs.
Okay, so the recent trends that are focused, I showed in my slides. They have the biofilm reactor and also the horizontal tubular reactor. The horizontal tubular reactor is one of the reactors which are currently being used a lot. I think the biofilm reactor is one of the recent trends which is currently being used in the industrial wastewater remediation, but can also be used in other purposes as well. And for carbon dioxide utilization, I think if we have better mass transfer and gas transfer, so if that problem is solved, then we can have then that is something we need to focus in our recent times.
Okay, yes, Miss. Thank you very much.
Okay, and then the next is Graha. Graha, you can go ahead for your question. Or the next is Alicia Nazwa.
Okay, thank you, Miss. Good morning. First, I want to introduce myself. I'm Asna Zualifiana from Pajajaran University. Okay, Miss Anggana, I want to ask you, from the mention of photobioreactor innovations like biofilm membranes, flat panel, street tanks, helix tubular, which is the most suitable for large-scale application and why? Thank you, Miss.
Um, so, which bioreactor is best for what? It depends on what product you are targeting on. So, like, for example, the helical tubular reactor is being used nowadays for production of like biofuels, and at the same time, the rotating reactors and the algal biofilms are being used for the industrial, bioremediation purposes. So it depends what your final target is. That would help us in narrowing down which bioreactor would be good for that purpose.
Okay, thank you, Miss Anggana, for the answer. Thank you, Miss.
Okay, and then another one is Hibane Konarno. You
Hello.
can go ahead for your question.
Okay, all right. Hello, Ms. Angana. I have a few questions. Currently, I am very interested in wastewater bioremediation using microalgae, especially in industrial wastewater like textile wastewater, is commonly characterized with high chemical and oxygen demand, which means it has a lot of heavy chemical components that could interrupt in biomass or algae biomass productivity. My question is it possible to produce high yield uh, biocomponents such as lipid or pigments using only that high chemical oxygen demand wastewater compared to uh, uh, using external nutrition to promote this biomass production. Thank you.
Yeah, so I would like to answer. It's a very good question, first of all, and I would like to answer this with like my personal experience. So, in one of my masters, my project involved like algae bacteria utilization of algae bacteria consortium in industrial wastewater for the production of single cell oil and calcium carbonate. So, what we did was we isolated a particular. First of all, the industrial wastewater which we had high contents of urea, so we had to select one kind of particular bacteria which can utilize the urea from them and produce calcium carbonate in return. So what we did was we made a consortium of the algae and the bacteria. So inside the algae and the bacteria, they kind of had a syntrophic relationship. Like the algae was taking in the carbon dioxide and the bacteria was giving out the carbon dioxide and the oxygen was being given out. So in this way, the same technology can be used in textile industries where some kind of other bacteria or even fungi or other kind of algae can utilize those contaminants. Then I think it can help this other my main microalgae to increase its biomass productivity as well.
So the algae could survive the high chemical content on that wastewater because of that bacteria consortium.
Yeah, I mean it was being able to survive alone as well, but the biomass productivity increased when the bacteria consortium was formed.
Okay. Thank you for your answer. I think in the future I would like to learn more about this, algae and bacteria consortia method. Thank you.
Okay, thank you, Hivan. And then the next is Widyarti Putri. You can go ahead for your question.
Thank you for the opportunity. Let me introduce myself. My name is Widyarti Putri from Pajajaran University, and I want to ask a question about photobioreactor sustainability. So the question is, how can photobioreactors be optimized to enhance the sustainable product of biofuels, considering the factors such as energy efficiency, scalability, research use, and the reduction of greenhouse gas emission while addressing challenges related to the economic feasibility of large-scale implementation? Thank you.
Yeah, so as I already discussed, like the energy, as you asked in the question itself, so optimizing the algae photobioreactors is important, and specifically keeping in mind the capital cost and the operational cost. So if we can reduce those the like the substrate or the media which in which the microalgae can grow in, it can reduce a lot of the operational cost in the beginning stages itself. However, you still need some technologies to separate out the single cell oil or the like the lipid bodies for the production of the biodiesel later on. But I think the most important thing is if we can optimize a microalgae which can grow in very high contaminant industrial effluents or something, then you can reduce the cost a lot and optimize the production as well.
Thank you, Miss, for the answer.
Okay, thank you, Widyarti. And then the next is Stephen C. Marmata.
Okay, thank you for the opportunity given. For the first, let me introduce myself. My name is Stephen Sumarmata. I'm from Pajajaran University, Agro-Industrial Program. So I want to ask about the growth of algae. How can we control the growth of microalgae if we do it in an open ecosystem such as maybe like a small lake, we compare to a photobioreactor. So I think maybe and what are the advantages and maybe disadvantages too. Thank you.
So yeah, I mean that is one of the drawbacks for the open photobioreactors. You cannot control the algae growth. That is why the innovations and the trends have moved the open photobioreactor to the closed photobioreactor. Whereas in the reactor, you can control the biomass growth. You can operate in a batch mode, continuous or a fed batch manner. That is not quite possible when you are doing in an open environment because it depends on everything like natural, the natural sunlight, natural light and contaminations are also there. So like it is as much as it is cheap in the process, but it also has some disadvantages like these. That's why the industries have also taken up the closed photobioreactor options rather than the open photobioreactors.
Okay, thank you for the answer, Miss. So does that mean we cannot manage it, right?
I mean, we can develop something which can be a hybrid between the open and the closed bioreactor, then that could solve the problem of the contaminations and, temperature control and everything.
Okay. Thank you. I understand now, Miss Angana. Thank you for your detailed explanation. Thank you.
Okay, thank you, Stephen. And then here, Martin Simanulang. You can go ahead
Okay.
for your question.
Thank you, Miss. Okay. Hello, ma'am. Good morning. First of all, let me introduce myself. My name is Martin Simanulang from Agro-Industrial Technology, Pajajaran University. I want to ask about photobioreactors. So we have talked about how photobioreactors can improving light distribution, but I kind of confused, like what technologies are used to achieve that, the improving light distribution with photobioreactors.
I didn't understand your question. Can you please repeat once more?
My question is, so we have talked about how photobioreactors can improving light distribution, but what technologies are used to achieve the improving light distribution with photobioreactors?
Oh, so you can use different kind of optical fibers or other like light measurements, which I am not sure exactly, but I know one of the ways is to use optical fibers, which gives them better light efficiencies. I can tell about only one right now.
Okay, ma'am. Thank you.
Okay, thank you, Martin. And then here, Raihana, you can go ahead for your question.
Okay, thank you for the opportunity. I want to introduce myself. My name is Rehana from Pajajaran University 2023. And I want to apologize for not being able to turn on my camera because there are any trouble. So the question is, in component NK design slide, it says their gas exchange system. Can this system be adapted from other gas, such as nitrogen or methane, depending on the application? And is there any explanation, or can you elaborate the gas exchange system in component NK design? Thank you.
Yes, so the gas exchange system, you cannot use the methane or other kind of gases because the microalgae particularly takes up the carbon dioxide gas. So it has to be a carbon dioxide gas. So like plants, like the plants for their photosynthesis, they take up the carbon dioxide and the sunlight for their process to produce the glucose and other things for them to survive. Similarly, the microalgae also takes up the carbon dioxide. So the gas exchange system has to be between carbon dioxide and oxygen. They release out oxygen, so there has to be a storage tank separately for the taking out of the oxygen from the medium, but it cannot be some other kind of oxygen, other kind of gas, because the but if all kind of microalgae and cyanobacteria takes up only carbon dioxide.
Okay, I understand. Thank you, Miss.
Yeah, thank you, Raihana. And next, Fauzia. You can go ahead for your question.
First of all, I apologize for not being able to turn on my camera. Let me introduce myself. My name is Fauzia Surya and I'm from Pajajaran University. So my question is, can you please explain what is light setting and temperature role in photobioreactor process?
With temperature and what you said?
Light setting, miss?
Light sensitivity.
Yes, miss.
Yeah, so so like I guess you have read previously about the bacteria also. So every kind of bacteria can grow only in particular temperatures, like either thermophilic or mesophilic temperature, or even at very like cold temperatures and all of them. So even for the microalgae, for them to work like in an optimal temperature and produce highest of the biomass, they need to be enough particular range. For example, Senedismus obliquus is one of the microalgae which grows in the range of 20 to 35 degrees Celsius. And there are some microalgae which can grow in very high temperature as well. And at the same time, the light sensitivity is also important because when you are operating in a closed photobioreactor, the whatever light it is getting, it is from an artificial method, like the one someone asked before, like how we can increase the light sensitivity. It can be by different methods, like it can be the bioreactor can be very thin as possible so that there is enough of light penetration, or you can change the lights to fiber optics and other stuff, or you can have it in an inclined way so that the microalgal cells can utilize most of the light in it. So the temperature and the light, both of it is important for the microalgae to grow and increase its biomass in the photobioreactor.
Okay, thank you so much for your answer, miss.
Yeah.
Yeah, thank you, Fazza. And then the next, Nazwa Dita. You can go ahead for your question.
Okay, thank you so much, Miss Aisyah. First of all, I apologize for not being able to open my camera because I'm currently feeling kind of a bit sick. And then let me introduce myself. My name is Nazwadita. I'm from Pajajaran University. And my question is, how can membrane technology innovation enhance biomass retention efficiency compared to other designs?
My membrane technology for the biomass efficiency here is very important. Like one of the recent trends and innovations about the photobioreactor is the membrane technology. So this membrane technology, like it can help in different stages, like in cultivation, harvesting, and in the downstream processing. Mostly in the downstream processing because it will need like separation through microfiltration, ultrafiltration, and forward osmosis. So whichever membrane technology we we may need. For example, if we are taking nanofiltration for separating out our components, so first of all we need one of the membranes to separate out the biosolids and the other microalgal cells present. So that is mostly done by the ultrafiltration techniques, where it can retain the smaller biomass particles. Now after that we can go for the forward osmosis process, or even like liquid extraction or other methods, of which can use other kind of membrane technologies. So the membrane technologies is mostly used in the downstream processing for these microalgal technologies. In a membrane bioreactor where they're using the algae, they are producing the algae, the like the triacyl glycerides or the single cell oil, and separating it out at the same time in the same place. So it might be an energy intensive, but still a little cost effective than the other methods.
Okay, I understand it now. Thank you, Miss.
Okay, thank you, Nazwa. And then next is Debrina Zarko. You can go ahead about for the question.
Okay, thank you, Miss Aisha. Let me introduce myself. My name is Debrina Zarka from Agricultural Industrial Technology, Pajajaran University. First of all, I'm sorry I cannot open my camera. So I will to ask you about types of photobioreactors. How is aeration applied in different types of photobioreactors to ensure optimal gas distribution? Thank you.
The optimal gas distribution depends how much your mass transfer, the gas transfer takes place. So for example, you can have a trickle bed reactor where there is a gas diffusion and better mass transfer as compared to a reactor like a continuous or a chemostat reactor. So it depends on how you are using it for agitation. It can be a fluidized bed reactor as well. Now, the fluidized bed reactor will help in better gas transfer or the diffusion of the gases. So that would enable them to have a better gas exchange between the microalgae in the bioreactor, and that would increase the biomass productivity.
I got it, Miss. Thank you.
Okay, thank you, Debrina. And then next is Michael. You can go ahead for the question.
Thank you for the opportunity. Please allow me to introduce myself. My name is Michael. I'm from Agro-Industrial Technology, Pajajaran University. So my question is there any major reason why we use photobioreactor? Because as we know, there are many types of bioreactors. So what is the major reason for using photobioreactor? Maybe you can answer it based upon your experience, Miss Angana. Thank you.
So I have not personally used photobioreactors. So most of my algal cultures I have done when I did my experiments were in flasks. But a photobioreactor is important because you need to scale up from the flasks to a higher level, right? So that you can do batch reactors or continuous reactor, and you can observe like ultimately your target is to scale up to a level where it can be used in the industries. So a photobioreactor is the best option, and we cannot use the other bioreactors mostly because the microalgae needs light mostly for their photosynthesis, right? You can get gas exchange can be done in the normal reactors, but a photobioreactor is specifically transparent so that the light, because of the light penetration to the microalgal cells. So the photobioreactor is important in the scaling up process, and at the same time because of the requirement of the microalgae.
Okay, thank you, Miss Angana. So the reason is because using photobioreactor is it can be upgraded to a higher scale.
Yeah.
Okay, thank you, Miss Angana.
Okay, thank you, Michael. And then the next is Nabila Putri Fadila. You can go ahead for the question.
Okay, good morning, Miss. I'm sorry I cannot turn on my camera due to a bad connection. My name is Nabila Putri from Pajajaran University. I would like to ask a question about open photobioreactors. One of the advantage mentioned is large-scale production. What makes open photobioreactors suitable for large-scale production?
Well, I wouldn't say it is suitable, but the open photobioreactors has a low operational cost as compared to the closed photobioreactors. because the open photobioreactors are not inside like a controlled parameter kind of a way, so it kind of works on everything natural sunlight, natural temperature, natural control. So it provides low operational and capital costs, but at the same time, I wouldn't say it is most suitable for the production.
Okay, thank you, Miss.
Okay, thank you, Nabila. And then the next is Albinia Treyonora. You can go ahead for the question.
Okay, thank you, Miss Aisyah. My name is Albinia Trianora from Pajajaran University. Sorry, I cannot turn on the camera because the signal is not good enough. I want to ask, are there specific industrial applications that have successfully implemented the design of open photobioreactors?
In initial days, the open bioreactors were used for mostly the wastewater remediation, because they are like in open raceway ponds or like circular ponds or things like that. But nowadays, the closed photobioreactors, like the rotating algal biofill photobioreactor, has been used for industrial remediation in a scaled up, in a proper, like an industrial way. Previously, the open photobioreactors used to be used for industrial bioremediation, wastewater remediation.
Okay, thank you, Miss.
Okay, thank you, Albinia. And then next is Nurafa Hermawati. You can go ahead for the question.
Okay, thank you, Miss. Let me introduce myself. My name is Norapa Hermawati. I'm from Pajajaran University. What is the most effective strategy to overcome the risk of overheating in closed system, especially in areas with high temperature or intensive lighting? How important is it to use materials that are resistant to change in temperature and heat in the design of closed photobioreactor? Thank you.
So in the closed photobioreactor, it's the microalgae responds a lot. It depends a lot on the temperature and the light sensitivity. So it's important that you use, you and at the same time you use materials which has enough amount of nutrients so that the microalgae can uh, use them to grow as well. It's not just the bioremediation that is happening is a by default mechanism. So what is happening is the microalgae is taking up the nitrogen and the phosphate or the other materials which is present in the substrate. So it's important that when you are supplying something which is not like a normal laboratory media, especially like a wastewater, it has, like, you have to be sure that it has high amount of nutrients in it so that the algae can actually utilize from it. And coming from the temperature perspective, so it's important to have an optimal temperature. Too much high of a temperature can kill the microalgae as well as it is for the bacteria. So microalgae behaves exactly like a bacteria or a fungi, so the temperature is very important to them, and it has a range in which you have to perform it always.
Okay, thank you, Miss.
Okay, thank you, Nurafa Hermawati. And then next is Magfira Akila. You can go ahead for the question.
Okay, thank you, Miss. Good morning, Miss Angana. Sorry, I can't turn on my camera. My name is Magfira from Universitas Pajajaran. My question is, how do different photobioreactor designs like the tubular, annular, flat panel, or the other designs affect their performance and application? Thank you.
The tubular, flat panel, and the other ones affect the efficiencies in the way how the microalgae is like positioned in them. And for all of these technologies have been been formed, or we have got these technologies like a step by step, probably you have one kind of bioreactor in the beginning, and then that needs to be innovated again, so they change it again to tubular photobioreactor, then again to a flat panel reactor. So the flat panel and all are like the solar plates, they have to be aligned in a particular angle so that the microalgae can take up the light in the, like, there is a particular angle in which the microalgae or in any plant can be placed, it can utilize the most amount of the light. Now, similarly, when you go for the other tubular reactors, for the tubular reactors, the algal cells get a higher amount of area for them to grow, and that will help them to produce more amount of biomass for the tubular bioreactors. Similarly, when you go to the next bioreactor, it has some other innovation or like some other improvement in it which can help in the biomass productivity.
Okay, thank you for the answer, Miss Angana.
Okay, thank you, Magfira. And then the next is Timotheus Andito. You can go ahead for the question, Timotheus.
Okay. First of all, let me introduce myself. My name is Timotheus Andito, and I'm from Agro-Industrial Technology, Pajajaran University, 2023. So I'm curious with like how is the pH level maintained in photobioreactors without negatively affecting microalgae growth?
Yeah, this is again like the microalgae is like the bacteria. So what if it depends on what species of microalgae you are using. For example, when I worked with a species of Ceratosmus, which used to grow in a pH of 6.5, 6 to 6.5. So it was little acidic as compared to others. But then when you have blue-green algae like cyanobacteria and all, it can grow in a range of 7 to 7.5. So it depends on what kind of species you are using, and so you have to optimize it beforehand before using it in a photobioreactor.
Okay, so it depends on the microalgae that we use.
Yeah.
Okay, thank you.
Okay, thank you, Timotheus. And then here, Graha Gunawan. You can go ahead for the question to Miss Anggana.
Okay. Good morning, Miss Aisyah and Miss Anggana. My name is Grahan Pragunawan, and I am from the Agriculture Industrial Technology Program Pajajaran University. As you have previously explained about the photoreactor, I would like to ask about what is the role of automate reactor design and genetic engineering in improving photoreactor efficiency despite challenges such as high cost, sub suboptimal challenge such in high cost and suboptimal light distribution and risk of contamination, and how to reduce the risk of contamination.
So, for genetic engineering is a very new technology which is being applied in every process. It's not limited only to the medical purposes. So, for microalgae also, there are two, like they can be used in solving the challenges at different stages. For example, one of the challenges is to produce the lipid bodies, like to extract the lipid bodies from the biomass itself. So we can genetically engineer the microalgae where it can produce a very higher amount of biomass, so that the lipid extraction is not only 40 limited to 40%, it can grow up to above 50%. And even for answering like the question of contamination also, generally microalgae grows in different area in different wastewaters. But in any case, if you want to grow it in a particular wastewater in which it is not growing, then maybe we can genetically modify it so that it can avoid other contamination and grow separately, and it's resistant to the other chemicals, or other the other chemicals which are present in the wastewater.
Okay, I understand. Thank you for answer to Miss Anggana.
Okay, thank you, Graha. And then the next is Giza. You can ask the question.
Okay, thanks, Miss. So, let me introduce myself. My name is Giza, and I'm from Agro-Industrial Technology of Pajajaran University. I have a question, like a really simple question, but I'm really curious. So, I want to ask, is there any genetic modification or enhancements used to optimize growth in photobioreactors? Thank you, Miss.
Yeah, they are doing, genetically modified microalgae where they can optimize them. They are increasing the biomass productivity and the amount in which, the wild strains has a particular level up to which it can take up the nutrients and phosphate, after which it cannot probably, or it is getting substrate toxicity. Too much of nitrogen or phosphate might be toxic for them. So we can genetically engineer the microalgal species where they can uptake even higher amount of the nitrogen and the phosphate for the growth and the production of biomass.
Okay, Miss. Thank you.
Okay, thank you, Giza. And then the next is Igede Wismaya. You can go ahead for the question.
Okay, thank you, Miss. Before I want to introduce myself, my name is Agri Maya from Agro Industrial Technology Class 2023, Pajajaran University. From what I know is photobioreactors can produce biofuel from microalgae. How effective is the biofuel that produced by microalgae in photobioreactors compared to other biofuel that produced by other substance? Thank you.
Yeah, so the efficiency of producing the biofuels from the microalgae is still less as compared to other biomasses. That is because we still have not yet have a technology which can utilize produce 80% of the lipid bodies from the biomass itself. So we generally reach up to 40 to 50 percentage of that. So I mean that is something which the scientists are already working on to see that if we can get some membrane technology or other methods where we can obtain the triacyl glycerides or the single cell oil which can further transesterified to produce the biofuels. So yeah, that is something which is one of the challenges that the scientists are working on.
Okay, thank you, Miss.
Okay, thank you, Igede Wismaya. And then next is Lutfiah Nur Aziza. You can go ahead for the question.
Okay, thank you, Miss. First, I apologize. I can turn on the camera because my connection was unstable. My name is Lutfiandro Aziza from Universitas Pajajaran. So my question is, how do photobioreactor handle issues such as contaminations during long-term operations? Thank you, Miss.
Yeah, so in open photobioreactors, even if for open photobioreactors, dealing with contamination is still easier. Like you add chemicals and then or otherwise replace the entire culture. Now for the closed photobioreactors, you have to be very sensitive so that you don't get contamination. And even if you get contamination, you have to use some limited amount of chemicals so that it can inhibit some other kind of microbes I mean, you can use antibiotics to prevent bacterial contamination if you want.
Okay, thank you for the answer, Miss.
Okay, thank you, Lutfiah. And then next is George Hasudunungan. You can go ahead for the question.
Okay, thank you for the opportunity, Miss. Before I ask my questions, let me introduce myself. My name is George, from Pajajaran University, class 2023. And I want to ask about the contaminations that you said that one of the challenges in photobioreactor is the contaminations, right? And
Yeah.
I want to ask, is there any technologies or is there any way to control the contaminations from, let's say, the wastewater? I think that's all my questions. Thank you.
Well, when it comes to wastewater, then it is already contaminated enough. You just have to pick a microalgae which can grow in a contaminated, like a wastewater, which can, withstand or resist the other chemicals which are present there. So that is only going to be the only other way.
Okay, thank you, Miss.
Okay, thank you, George. And then the next is Gina Mutia Zahra. You can go ahead for the question to Miss Angana.
Okay, thank you for the opportunity. First, let me introduce myself. My name is Gina. I want to ask, in the context of agro-industrial technology, what are the advantages and challenges faced in applying photobioreactor technology to produce environmentally friendly bioplastics? And how can the use of agro-industrial waste and agricultural residues be maximized in the microalgae production process? Thank you.
I didn't get one was the plastic bag, and what was the other bioreactor that you asked?
I'm sorry, what?
Like you asked one about the plastic photobioreactor. What was the other one that you asked for?
How can the use of agro-industrial waste and agricultural residues be maximized in the microalgae production process?
Okay, so like agricultural residues. So the algal biomass can be utilized in producing biofertilizers, biopesticides, and at the same time the single cell oils as well. So I mean, you can use different kind of bioreactors for the different purposes. Now that plastic biofilm is the plastic bag bioreactor, it might be environment friendly, but it will have different kind of challenges like the mass transfer limitation and the gas transfer limitation. Since it's a plastic bag, it is not going to be hard enough. So there are chances that there might be leakage or other contamination, and at the same time it might not be able to hold up the entire algae reactor. It can be used for a homely product or something, but for large scale production of the algae for the agricultural residues or other something of that sort, I don't think the plastic one will be good enough.
Okay, thank you for your answer.
Okay, thank you, Gina. And then the next is Rizwan Nurhalisa. You can go ahead for your question to Miss Anggana.
Okay, thank you for the opportunity, Miss. My name is Risma Halisa, and I'm from Pajajaran University. I would like to ask, how does the size of a photobioreactor can influence its energy consumption and overall efficiency?
Um, uh, the photobioreactor for the energy efficiency, we can deal it depending on the what kind of photobioreactor we are trying to use. So if we can reduce the cost or of the initial substrates, like using wastewater instead of other media, or choosing a microalgae which can grow in any place or any kind of media, then we can decrease the cost parameters. And at the same time, if at the same time, in the separation technology as well, we find some techniques like other than the membrane technologies which can decrease our energy efficiency. Then that can help us in the reduction of the energy efficiency for the entire process.
Okay, I'm understand, Miss. Thank you for the answer.
Okay, thank you, Risma. And then next is Stefan Simarmata. You want to ask again to Miss Anggana?
Okay, thank you for the opportunity, Miss. So here I want to ask again, in the biotechnology industry, the production of high value compounds such as pigment and fatty acids is often a priority. However, the growth of microalgae in photobioreactor can be influenced by competition between biomass production and target compounds. So to use genetic engineering to direct microalgae to focus more on the production of high value compounds without sacrificing biomass growth. Thank you.
I think you were cut off in the middle, but as much as I understood, you wanted to know like how we can control the competition between the biomass productivity and the compounds, right? So
Yes.
so the compounds that you are going to get are going to be extracted from the biomass itself. Now it depends how you are going to extract from the biomass by different technologies. So it's not really a competition between the biomass productivity and the other compounds. Anything that any high value compounds that you are going to get is from the biomass itself. So I mean, that's not really a competition. The more the biomass, the more it will help you in obtaining the high value compounds.
Okay, thank you. Thank you for your explanations. Thank you.
Okay, thank you, Stephen. And then next is Mahesa. You can ask the question.
Okay, thank you for the opportunity, Miss. Um, First of all, let me introduce myself. My name is Mahesalia from Pajajaran University. So it was mentioned earlier about the advantages and disadvantages of post-photobioreactor, one of which is falling and maintenance issues. So the question is, are there specific material used in the design of post photobioreactor that can help reduce the risk of harm?
Use of the different materials, like I don't know if you're asking about the material of the photobioreactor or the substrate, but yeah, I mean, for the photobioreactor, since it's algae used is highly light sensitive, so the material used has to be something which can help in the penetration of the lights. And for the material for the substrate, as cheap as the substrate it would be, that would be better for the cost of the entire project.
Okay, thank you, Miss, for the answer.
Okay, thank you, Mahesa, for the question and Miss Anggana for the answer. And then next is Nazifa. You can go ahead for the question.
Okay, thank you, Miss. Hello, Miss. Let me introduce myself. My name is Nazifa Kapela Zahra from Pajajaran University. I would like to ask, this is possible to use photobioreactor to process mycositis alga into fertilizer or as substitute semiconductor material to replace nickel. Considering that the mycositis alga is toxic, could this toxicity affect plants if used fertilizer? And conditionally, can a photobioreactor be used to remove the toxins during the process, or any bioreactor remove the toxin? Thank you.
I didn't understand where you want to use the semiconductors or the nickel. So I mean the algae there might be a toxicity in it, but I don't understand where you are trying to use the semiconductor for, inside the bioreactor or outside.
But photobioreactor remove the toxin in the fertilizer or not, Miss?
Okay, the photobioreactor and help. Okay, so for heavy metals, I still have not found any paper or any work where they are able to remove heavy metal compounds. They are good in removing nitrogen, phosphate, and other stuff, but for heavy metals, I don't think. I mean, you can genetically modify something, some microalgae, but I don't know a wide strain which can take up a heavy metal yet.
But because sorry, Miss. But mycositis alga is not in photobioreactor remove the toxin in mycositis alga or not?
I have to look into that. I'm not sure about it.
Okay, Miss. Thank you.
Okay, thank you, Nazifa, for the question. And maybe there were many questions from participants showing a high level of interest in photobioreactor, but maybe other question can be written in the link that I will be sent, and later I will send it to Miss Anggana via email, and Miss Anggana can answer the question, and I will convey the answer from Miss Anggana to the other question. It's okay, Miss Anggana, if
Yeah.
we do
Sure,
that?
sure. Yeah.
Okay. Maybe so many participant interesting in the photobioreactor, and then maybe thank you very much for your detailed and insightful answer, Miss Anggana. Uh, sorry, I will send the link.
Yeah, sure. Thank you for the invitation. It was a good talk. Thank you.
Yes.
See you, everyone.
I think Aisa has the certificate
Yes.
for you too, right? We want to show the certificate for Angana.
Okay.
So one more time, thank you so much, Angana, for today. So I'm so sorry. I actually just I joined it and then I left, but that's so interesting topic. A lot of questions today from the students. It means like your presentation is really good, and then I really hope that the students here enjoy and you also enjoy to do presentations today. Thank you so much, by the way, Angana.
Yeah, okay. And then as a token of our presentation, we would like to present you with the certificate of appreciation. Muklis, can you share screen the certificate? Okay. Thank you once again for your exceptional contribution and for sharing your valuable time with us. And we look forward for more opportunities to learn from you in the future. Thank you, Miss Angana, and thank you everyone for your active participation. So have a great day.
Yeah, for today is the last international lecture series. So I actually would like to say thank you to everyone, especially for the speaker, from the first speaker until now. Angana, thank you so much for your effort and then for your time too. So yes, probably next year, I and the Department of Agroindustrial Technology, we also will make this the same events like this probably next year. So thank you so much for coming, and okay, see you next year probably. Okay, thank you. Bye bye.
Thank you so much, Mr. Budi and Miss Aisyah.
Thank you so much, Mr. Budi and Miss Aisyah.